Hi, everybody. Welcome to The New Normal. I'm your host, Lisa Davis, aka like Queen Lisa. Today is my very dear friend, Dr. Reg McDaniel, joining us to try to help everybody with your wellness, your health, and bring it all back to our bodies healing ourselves. So he'll be right back with us right after this. Welcome, Dr. McDaniel. Thank you for joining us again. I am so glad to see you. I have several friends now taking New Eden on a regular basis. We have somebody in the comments. I wanted to, just before we get started, have you answer just one quick question. Michelle Michaela says that she started taking New Eden when we did the last show. She's taking 3 scoops in the morning, 3 scoops in the evening, using unsweetened organic coconut milk. She has some lung issues. And could you ask if that is okay?
Yes, that's quite good. Particularly dividing it in the morning and at night is good. And of course, coconut milk is good stuff. I
Okay.
feel like it in pina coladas.
You can even add a little rum to that, Michaela, and or Michelle Michaela, and maybe that will be good for you. So that's great. And it's great to start off with such a strong beginning like that, because she'll really get a great concentration of nutrition to be a great start for her. So that's wonderful. You did want to give us kind of a preface to this first video. We're going to break it up into 2 sessions. It is a little bit long, but it's a very controversial subject. And so can you give us a little bit kind of guidance or idea about what this next video is about?
Well, I feel very uneasy about this presentation because since 1994, I have tried repeatedly to be more and more simple if possible because many of the scientific terms and medical terms are in Greek syllables and Latin syllables, and we don't talk like that every day, and it's hard for people to follow scientific and medical information. However, I was 87 years old last week, and I have never lived in a period when there's more controversy and hostility and disagreement going on, and even among experts that some have influential university and government positions, and others at other universities and maybe farmer or government positions don't agree with them. And surely the public is wondering why. And I'm going to try to the best of my effort to tell you some of the reasons that such conflict is going on between what you would think would be people that were like science is supposed to be the search for truth. And whose version of truth are you supposed to believe? Well, a lot of conflict is going on. For example, there was the Barrington Proclamation that was signed by, if I recall correctly, about 10,000 scientists and medical people from 40 different countries. And as more and more people have heard of it, it's nearly up to 1 million people that have signed protesting policies and mandates of the pandemic. And as it's expanded, the people are also wanting to expand it to this going green political movement that's going on. And put it simply, as I point out, is these scientists look for things that support whatever their agenda or position is, and that's what they tell you about, and the media carries it. And they ignore data or information or knowledge that doesn't support what they're saying. Well, the people in the Barrington Proclamation are looking at things that have been historically well documented that current politicalization of science and medicine has caused a conflict. And unfortunately, there's this thing going on that anybody that disagrees with the media or the political party in power is ignored or silenced. And so balanced discussion is prevented. Well, I'm going to try to the best of my ability, and I wished we had an audience and could periodically somebody, do you follow what I'm trying to tell you? Do I need to be a little more obnoxious? But
I
we'll
I
take off.
follow.
We'll take
Yeah.
off now and see if uh, what I'm trying to do is of any value to the layperson who wonders what in the world is going on.
Yeah, we're taking a little bit of a risk here. I hope and pray I don't lose my channel for talking about this. Um, but we're going to be bold and we're going to tell the truth. And this is a doctor, Dr. Reg McDaniel, who is giving his scientific information that he's sharing with us. So we'll go from there and we'll share the video. And we're going to do, like I said, about half of the video this week, and then we will come back with more discussion next time to share the video with you guys.
This lecture that I'm going to attempt to share with you, by its very nature, violates everything I've been trying to do since the dietary supplement industry was founded in 1994 with the passage of the Dietary Supplement Health and Education Act. And the reason I'm doing it is because surely with various publications, television, on your cell phones and computers, you're getting all this information about conflicts that are recurring with people charging that there's been politicizing of scientific information, and that people have agendas that they support any data that they can find that supports that agenda, and they ignore any information that doesn't support their agenda for what they're trying to promote. I hope that you'll stay with me because, for example, I love to go to the symphony orchestra in Dallas or Fort Worth. I can't play an instrument on the amphitheater. I can't read music. However, I can appreciate the music. And I hope that as we go through this information that you may not understand or comprehend everything that's quite detailed that will be in this lecture, but I hope you can hear the music of how incredibly detailed the process of life, of where the genes in each of our cells take elements on the composition of matter chart, which is about 100, and there is absolutely no evidence or sign of any kind of life in any of these elements, yet our cells with the code that is in nucleic acids or our genes or DNA can take these elements and hook them together, and behold, we have life. And we'll go through some of this process that I hope, again, that you'll understand or hear the music. But much like when I go to the symphony, I can't play any instrument or could not read any of the music that produces the sounds and harmony and beauty that comes from music. So with this, we'll start with the first slide. You may or may not be aware of that in the year 2020, there was an open letter published that 10,000 medical and scientific individual professionals signed the Barrington Declaration. Now, this was carried primarily in the conservative media, but the national media pretty well dropped it because they did not want the attention brought to the fact that these thousands of scientific and medical professionals were protesting the politicalization of the coronavirus pandemic, and also details about the problems of the RNA shot and various pandemic measures of 6 feet and everybody wearing a mask and other things that in the past that scientifically had been shown that they didn't do much. And here they were enforcing this with mandates, demanding that people do it, and the public media has been promoting this. With time, this proclamation is the efforts have been made to kind of expand it to include the politicalization of climate change. Now, there's no question that the climate's been changing as long as I've been around, which will soon be 90 years. But saying it's due to industrialization or the efforts of man becomes a very interesting question. And as I'll point out, those people that have questioned this because it's not part of the political agenda have been penalized. And this Barrington Declaration now, if you check, it's close to 1 million scientists and medical people have signed it, all protesting the politicalization of scientific information. And they sick and tired of, quote, the political agenda or the story of the political party in power and the media that seems to be their spokesman are developing an agenda about the pandemic and also about the climate change, and penalizing the oil and gas industry and anything that's source of energy of the past, and promoting solar and wind energy. So we'll go to the next slide. A good example of this, you might say, well, does politicalization of science matter? Well, I was watching television, and on one of the conservative channels, they brought out a segment that a community college professor that had been teaching biology for 20 years in San Antonio, Texas, in his lecture on chromosomes, showed on the screen that in the 23 chromosomes, chromosomal pairs, there's a total of 46 chromosomes, but they're in pairs, that in the 23rd position, if you have 2 X chromosomes, that's the karyotype for a female. And all the characteristics hormonally and physically that we associate with females are associated with this XX pair of chromosomes in the 23rd position. However, he also showed a karyotype, which is all the chromosomes laid out. I'll show you an example here in a minute. That an XY, and little y is just a tiny portion of the X, that if you have an XY, this is the karyotype of a human male. Well, 4 students in this lecture stood up, walked out, filed a protest with the university administration. And what was their beef and what is their agenda? Well, they said that sex is determined by one's feelings and socialization and personal preference. And this is what produces males and females, which is the agenda that's being promoted across the country in education. Well, allegedly, the teacher was fired for promoting religious dogma. And they ignore the science that was presented, that's been available for decades. In this case is on appeal. So this is just one example of how ridiculous politicalization of science can become, where truth is trashed, even when it is well-established scientific fact. Now, this is what the professor showed. And to the right, you see a jumble of chromosomes from one white cell where you have added a fluorescent dye so that it binds to all the nucleic acids or the DNA, and they show up. And you take a photograph of this jumble of slides from one white cell, and every cell in the body has this in the center, and you lay it out to the left from the longest in the number one position to the shortest in the 23rd position, which you see in the yellow block. And this is the karyotype of a human male. If you had two X's identical laying side by side, it would be a female. This has been known for decades. And I show it to you because this is hard, well-established science that by political agendas, this professor got fired, and one can only hope that out of this legal conflict, the truth will prevail. Now, the effort to expand from protests about the coronavirus, then where it came from, and the problems of the RNA shot. And I will not call it a RNA vaccine, which I will get into and show you why. But they've tried to expand this, that there is politicalization of climate change. And in this, the geological history of the continents before the industrial age is ignored, of times of when it became very dry and wet, and times very cold and very hot, totally unrelated to the Industrial Revolution. And that the fact that people that are promoting this agenda look for data and sites where they collect information that supports their propaganda or their agenda, and they cite this. However, there are data can be gathered from sites that do not support this agenda, and those are ignored. And in fact, a professor at the University of Miami and Miami, Florida, published a paper pointing out this and giving a balanced presentation. And his peers caused so much furor that buy into the agenda that he lost his position on the faculty at this university. So this is not just a theoretical thing. This has actually happened. And what it is that open and balanced debate is silence, not only on the pandemic, but on the climate change and on the new form of the vaccine type technology. And out of this, there are actually federal mandates demanding that citizens do certain things and don't do other things. And in fact, the oil and gas industry has been terribly penalized. And we've gone from a country that export energy to one that has to import energy because of federal policies against oil and gas industry. Even gotten into major sources of funding to block loans for companies that want to develop oil and gas. And of course, the price of gasoline has markedly been increased by this. But as you look on the highway and see the number of trucks that are absolutely essential for moving goods across the country, that God only knows what's going to happen with the supply of diesel, which these trucks burn huge amounts of, and it comes from the oil and gas industry. And of course, the solar and wind industry is being promoted with tax funds, is because that is positive in this agenda that we're living under. Now, the coronavirus 19 issue is more complex, and that's where in this lecture that I'm afraid that the details of it are because so much in science is based on words and phrases that come from Latin and Greek, and we don't use them in common everyday language. And so the layperson that has not had, say, multiple courses in biological and scientific science don't know what you're talking about. And that I want to point out that Robert Redfield, a medical doctor who was head of the Communicable Disease Center, made a statement early in the pandemic, and I quote him here: "I have analyzed the viral nucleic acid sequences," and that's the DNA in the virus, and that the changes from the wild strain to the pandemic strain do not reflect a natural mutation. He went on further to say that the DNA sequences are more in keeping with human manipulation. And for this statement, he was silenced, and the national media just dropped it like a hot potato because they didn't want an expert that had been in the field of virology for decades questioning the source of the virus that may or may not have come from the Wuhan laboratory in China. Also, you have found it science that has been suppressed, that a former Wuhan lab technician said, "We worked on contract with natural viruses on an American NIH, National Institute of Health funding grant." Now, this was initially denied by Dr. Anthony Fauci, but under the Freedom of Information Act that allows people to go into the records of the federal government, it was found out that this was a bald-faced lie, and that hundreds of thousands of dollars had been given to this laboratory in China, because I won't get really much into it, because the NIH has been financing, and the Department of Defense over the years, different projects for biological warfare. And under the Freedom of Information Act, this has been uncovered. And so they've gone to great lengths to try to find ways to do the research they want to do without being able to be understood or found by the citizens of the country that are actually paying the taxes for this many people feel like onerous activity. But there was no question that they financed research to alter wild virus genes found in animals in this laboratory under an NIH grant. Now, because of the problems that have come from the RNA technology, which I'll briefly get into, that for over 50 years, the Center for Disease Control had a definition, and I'll quote to you, quote, "A vaccine is produced by a substance that prevents an infection." Well, this new technology doesn't prevent an infection. So for a politicized definition, the CDC changed the definition of a vaccine to a vaccine is produced by a substance that stimulates the immune system. And I might add, the evidence to date strongly suggests that it confuses the immune system with this new technology. And I hope before we're through, you will understand in the process of how cells work. I had 2 years working on an academic degree in cellular physiology, which is a big words to say how do cells work. Individual cells in the plant kingdom and individual cells in the animal kingdom, of which human beings are a part of. And we have trillions of cells, and they all have to work together, and they all have incredible systems in them that time and time again, I have asked, how in the world did this ever come to be? So detailed and such design. And of course, there are those with an agenda out there in the media and in education saying there is no intelligent design. And I'll leave that up to you as we go through some of the, just some of the information about how cells work that I think is very important to know, to understand or comprehend how there could be so much controversy and the issuing of the Barrington Proclamation of scientists and medical people protesting the politicalization of science. Now, let's focus a little bit on the RNA technology. And what is the RNA? Well, desoxynucleic acid, or DNA, is the code of life in plants and animals. And I was surprised to find out that the DNA in plant genes is exactly the same DNA that is in the animal kingdom. And that raises a question, how did this come to be? And I won't go into the experiments that have been done to show that you can insert certain gene segments from the human genome or karyotype that I showed you before into the plant kingdom, and that proteins and enzymes can be made by the plant that can be extracted, and they're just exactly like the protein or the enzyme that is made in the animal cells. Now, in the desoxynucleic acid, there are 4 nucleic acid molecules that make up DNA. And we use an A for adenine is one of them. And then we have a T for thymine, which is the second amino acid. And the third one is guanine, which we use G to, in the writings to keep from having to spell out the word every time. And then cytosine, which is the fourth one. Now, the importance about this 4 amino acid molecules is that this code is much like what you hear about. There is a code in your cell phone or your laptop or your desktop computer, that this code is somewhere between 10 times and, shall we say, 1,000 times more powerful than the code that is in this cell phone right here that I hold up in my hand. And one of the reasons I know this is because of when you have a little success in science, there are always those that think you know a whole lot more than you do. And here in the Dallas-Fort Worth area, there are actually research labs trying to develop software and hardware will be the computers we'll have 5, 10, 15, 20 years from now. And I've had them come and spend a half a day with me wanting to know, thinking I know everything, how in the world an individual cell works at a temperature that we are about 98/16 degrees. And we have a cooling and heating system of our body that dissipates, keeps the temperature of the body at a pretty narrow zone. And it takes very little energy to run this computer code, which is part of, I would say, the symphony of life that I hope that you are beginning to appreciate, what I call the music of life, even if you don't follow all these details I give you. Now, but the DNA code is, as I say, the code of life, and it's in the nucleus of every cell, and there's a nuclear membrane around it. And the DNA can be considered much like the reference books in a library. If you had any education, you know that you may want information or going to have a test. If you go to the library, you cannot take the reference books out of the library, but you can make a copy on a copy machine, and you can take it to your office or your room or any place you study or work. And in our cells, a copy is made from DNA inside the nuclear membrane, and this copy made is called RNA, and it's a little change in the sugar from desoxy to ribose sugar with these adenine, thymidine, guanine, and cytosine. And in the RNA, the cytosine is replaced by another molecule called uracil. But the RNA is a working copy that goes to the assembly line that is in every cell that carries on cellular synthesis of hooking together nutrient molecules to make bioactive compounds that run the biochemistry of life. And this is true for every system in the body, not only the brain function, not only the cardiovascular heart system, not only the hormonal system, not only your kidneys and GI tract, but this is true for all systems. And these nutrient molecules that are hooked together start with amino acids, and then there are the carbohydrate units that are saccharides or sugars. And then with this, we have vitamins and minerals and very special plant-made phytonutrients. That's not FIDO, but PHYTO, phytonutrients, the molecules that when you hook them together, you get a biological compound that has taken elements that have absolutely no properties of life, and behold, we start seeing the aspects of life. Now, when RNA is made from the DNA, there is a very special system where you have a adenine in DNA. In the RNA, you will always have a thymidine that hooks together or is made from that. It's called a complementary copy, where you have a thymidine in a DNA chain, you'll have adenine in the RNA copy. And as you go on down, you'll see where you have a G that guanine always attaches to the cytosine. And the remarkable thing about this is that I have them laid out in a straight line there. And oftentimes you see a helix in magazines or pictures when they show you chromosomes. Well, at 60 degrees temperature, you have a helix form where the maternal DNA sequence is wrapped around the paternal nucleic acid history of the male, and the combination of the male and the female egg and the male sperm. And this is very intricate in terms of that the adenine and the guanine are one class of molecules that I'll show you called a purine, and that the thymidine and cytidine are called a well, I'll just show you with the next slide. I believe it is. Well, no, before we get there, that they have instruments that you put in a buffer the free amino acids with a high temperature polymerase, which is an enzyme that is used to connect these nucleic acids in the order that I showed you before. But at 100 degrees, the helix unwinds and lays out flat. And with the polymerase, the nucleic acids can bind complementary, as I showed you before, to the chain. And then you go back to 60 degrees and it goes back to a helix. And so where you had one copy of each side, now you've got two helix. Then this little machine for polymerase chain reaction goes to 100 again, and the chromosome chain unwinds and lays out flat, and there the polymerase enzyme and the free nucleic acids can complementary bind again, and you go back to 60 degrees, and where you had two, you now have four. And this is goes over and over again, 60 to 100, 60 to 100. And as you see out there, it goes, it doubles every time. And where you have only one chain in a sample, if this goes all night long, going from 60 degrees to 100 degrees, you can have 1 million target nucleic acids there that can be analyzed. Now, there's a big problem with this, and you may have heard in the media that the person that developed this polymerase chain reaction said that the tests for the coronavirus that utilizes the polymerase chain reaction is fallacious. It cannot be used. And the reason is that if you had more than one chain, let's say you have maybe 6 different chains of viruses, the next morning you will have 6 million different targets all in the same product, so that this little chain of a virus that goes through this process of doubling every time the temperature is changed, doubles it. Well, everything in there doubles. And so all of us have in our serum or in our cells multiple viruses. And so you don't have a pure specimen to start out with. And this gives you false information in terms of trying to determine if a person has the China virus or doesn't have it, because there is so much reproduction through this polymerase chain reaction. Now, the RNA made from the DNA is divided into 2 functional types. And one part of the RNA is processed in a little area of the cell called the nucleolus, and it becomes what is called transfer RNA. And you put a little T in front of the RNA to stand for transfer. And of all things, this RNA goes into the cell cytoplasm, the fluid inside the cell, and in the interstitial space between cells, and looks for and finds to and binds nutrient molecules that will be required in the assembly line to make the bioactive compound that the other part of the RNA, which is called mRNA, which is messenger RNA, that carries the instructions to the assembly line that's in every cell to tell it where and how to put the different nutrient molecules that come from the transfer RNA that are brought in right at the right time, the exact nutrient that is recalled for in our genes. And I might add that you probably read that there are 30,000 genes. Well, depending on which one of those genes is activated to be expressed is what this little assembly line in the cell makes. In other words, it may make hormones, it may make neurotransmitters, it may make structure or functional compounds that are used in the body and used in the cell, or can be put into the serum and pumped around by the flow of the blood to the organs where it's needed. And I hope, though this is rather detailed, that you're beginning to appreciate that there's absolutely wonderful music in the organization and design of life, of how the nucleic acids work and function, and that this is solid, well-established science that has been established over the last 50 to nearly 100 years, that slowly, multiple laboratories in different countries have come together and published. And then not only do they accept it doing one, you've got to do it again in another laboratory to prove what was published in the first paper can be duplicated before there's acceptance to determine that this is hardcore, well-established scientific information. And that's where the Y chromosome and the two X chromosomes that I showed in the original karyotype came into being, that this is the karyotype for a female and the karyotype for a male, and that it's all in every cell of the human body, either the double X for the female or the XY for the male. Absolutely incredible detail, design, and engineering. And this is just to emphasize that in each cell, there is an assembly line, which in this cartoon that's derived from a paper in Annual Reviews of Biochemistry, 1985, by a husband and wife biochemistry team that has over 300 references to published papers, that they put it all together to show you that in every cell there's this assembly line that I have big letters 1, 2, 3, which you don't have to recall. We're not going to have a test on this, but the first step is the ribosome, and primarily there, the RNA strip, the messenger RNA, the little mRNA, is read to tell you the order in which amino acids are put together to start a little chain of amino acids called a peptide, which is on the way to become a protein if it gets high enough molecular weight. Well, it goes to the second step called the endoplasmic reticulum, which is a membrane structure. And here the first carbohydrate is added to it. And what's the importance of this is the biochemistry of what is happening is in the center. And you see some round circles, and it looks like a little pitchfork there, that it's 3 chains, and it's 9 molecules of mannose sugar. And the reason I have that there is because that is the principal molecule that is used in cellular synthesis that comes from the aloe vera leaf gel, mannose, acetylated mannose. And this makes a domain, which you can see on the surface of the cell over there to your left. And there are many of these on the surface. In fact, we just have a few of them there because it gets like shag carpet and you can't even see them. But this domain is for cell-to-cell communication, because the trillions of cells in the human body must work together in harmony. And that's what this domain, made from the principal carbohydrate unit that's in aloe vera leaf gel, is assembled there in the second step of the endoplasmic reticulum. And then it begins to be modified as you go around, as it goes to the Golgi in step 3. And at the bottom of the cartoon, you have all the structure and function types of molecules that make up what this little assembly line can make inside of every cell. And before I leave this, I should point out that the cartoon of the cell that shows the nucleus and the DNA inside the nuclear membrane is what you can see on a light microscope at about 1,000 magnification. Well, it looks like in the cytoplasm around the nucleus, there's nothing there. but with proper stains and electron microscopy up to about 100,000 magnification, you can see the ribosome, step one in synthesis, the endoplasmic reticulum 2, and then the Golgi apparatus membranes is number 3. That is not the same magnification. That on 1, 2, 3 is about at 100,000 magnification. The cell to the far left is about 1,000 magnification. And of course, the activity inside of the biochemistry going on. And again, I'm trying to point out that the dynamics, engineering and design that goes on in every cell in the human body follows this pattern. And if you understand this diagram, you understand the power of nutrition, because depending upon which gene is activated out of the 30,000 genes, the biochemistry in the center changes. There will be different amino acids, different fatty acids, different carbohydrate units, different phytochemicals from the plant kingdom, minerals and all to make up whether it's going to be a hormone or an enzyme or a structure in the human body or a functional molecule that's important to know that that code that is in the karyotype that we started out runs all of this and is the information. And so the RNA that was made from the DNA is run through pulled through the ribosome to tell you which amino acids and how they're to be hooked together. Also, I attended a course in Washington, D.C. for a week to try to understand as much as possible about this. And from this point on in this course, they said downstream processing, because the details of how the biochemistry is modified that is in the center is really not well known. There are many papers being published trying to determine the entire process of going from a molecule that has no properties of life to be hooked together to make bioactive compounds that carry out biochemistry of life in our body. But again, I don't expect you to understand or comprehend all of this, but I do hope, like I was talking about going to the symphony, that you appreciate the incredible music of the symphony of life that I'm trying to lay out to you. And we're just barely getting the basic information started. Now, this is a model of the cell, and that the energy to run everything in the cell, like that assembly line, comes from mitochondria. And also, you will notice a little red thing under the nucleolus, that the RNA that is made from the DNA, that is the transport RNA to go out and look for the nutrient molecules, is processed in a little area inside the nucleus called the nucleolus to become the transport RNA. And the cartoon had everything laid out in a kind of assembly line, but it's all kind of jumbled up in a cell. And remarkably, the cell knows how to do all this. And of course, the mitochondria is providing the energy that from breaking the bonds of sugars and the between each carbon bond, which in the process of photosynthesis, where energy comes from the sun to hit chlorophyll, and with chlorophyll and carbon dioxide, which we breathe out and which comes from fermentation processes, these carbon atoms are hooked together, and a little bit of the energy from the sun is put in that bond between carbons. But the mitochondria reverses this process of energy collection from the sun and transfers a little bit of that energy to ATP, adenosine triphosphate, which is the wet cell battery that everything in the process of life is run on that energy. Also, there's some incredible information that these mitochondria are much like what happens with your hot water heater in your home or your central processing unit for heating the house. It generates free radicals and nascent oxygen, which try to attach to anything they can find that it's like a magnet. If you've got a north and a south magnet, how they attract each other. Well, these free radicals will, in the design of life on that assembly line, the body makes glutathione and superoxide dismutase, which are protective, and it's like a little cloud around each mitochondria to soak up or bind these free radicals to keep them from attaching to membranes in the cell, or even worse, to the nucleic acid or the DNA that's in the chromosomes of the process of life, because the order in which the molecules are put together in the DNA chain has information. If anything new is attached to it, like one of these free radicals, it changes the information. And the worst thing to do is called a mutation, which can end up being cancerous, that the cell no longer respects all the other cells and doesn't work together in cooperation. And these malignant cells will not honor the existence of other cells and try to take on their nutrients and occupy their space. And of course, malignant cells can actually cease the ability to be alive and cause cancer death. Well, this is just a little bit about the symphony of life and what goes on in one of the trillions of cells in your body, and how DNA is transferred to make copies that are RNA, and that there are two kinds of RNA, and that they function differently. Now, I was mentioning about the glutathione and superoxide dismutase. This is a slide given to me from Texas A&M, where they have a very special microscope that you have living cells in culture, and that what you see there, where you have in each one of them, is the nucleus in liver cells. Well, these cells are scanned at different wavelengths, and then they can make a composite picture of what glowed at the different wavelengths of this microscope that stores this information in the computer. And what they do is they put a antibody in the media that will attach to glutathione and to dismutase. And one of them is green, which is the glutathione, and yellow is the superoxide dismutase. And in fact, there's a little cloud of glutathione and superoxide dismutase around every mitochondria to try to soak up these free radicals before they can attach to anything else in the cell. And again, this is more detailed than I think that you generally would be interested in. But just like I say, going to the symphony, I appreciate the music, even though I can't read music and can't play any instrument on the stage. I'm trying to get across to you how beautiful the design of life going on in each cell in your body, and how it's detailed, and how it works, and why scientists are protesting people that know nothing about all of this come up with things like what is a male or a female is whatever you want to decide to be, rather than how your chromosomes came together at the time of conception. Now, this is an incredible display of truth. about in the center with the big round center there to the left, you see the nucleus. And what we have here is that the transport RNA, and this is a very special preparation where a silver layer was placed over the cell with radioactive transport RNA, giving off energy that's radiant energy, and it exposes the film and makes it black, just like on a mirror or in a black and white photograph. But it's all going on inside the nucleus. And then after it becomes transport RNA, it diffuses out in the cell, as you see to the right, looking for the nutrient molecules to bring to the assembly line that I have shown you. And this slide on the right, 88 minutes after activation of the gene, you can see that the T transport RNA is looking for the nutrient molecules in the fluid inside the cell that hopefully in your diet you have provided the nutrient molecules that are required on the assembly line to properly assemble the bioactive compound. And I have a question mark there because it looks like that the nucleolus, which is the high processing area for the transport RNA, has moved out into the cytoplasm and is still making RNA. And that raises a question about what we do not know, but only this is done because of silver deposits by using isotopes that release radiation and expose the where the molecule is. And you can tell, and this is proof that the transport RNA is processed in the nucleolus and then moves out looking for the nutrients to bring to the assembly line that I've shown you. Now, the RNA, first thing I told you about was the information to tell the ribosome how to hook the amino acids and which ones together to start making the peptide or protein. The carbohydrates are added to the peptide in the endoplasmic reticulum in step number 2. And the number 3, the Golgi, is kind of like the finishing stage of an assembly line. And in Russian research behind the Iron Curtain, they were able to show that the trace elements, which are found in humate and are so low that you have to have a large amount of cells to even detect the trace elements, but the trace elements measure up to about 70, very small amount. And a good example that maybe you know about is that hemoglobin does not work or carry oxygen without its iron molecule at the center of the amino acids and the fatty acids and all, making a complex hemoglobin molecule, but you got to add the element. And this is a principle that's repeated time and time again. Now, I want to point out that the RNA quote unquote vaccine, which I want to call a shot because they had to change the definition because what they were doing doesn't prevent an infection, it just stimulates the immune system. That was a brilliant idea. And the reason it was a brilliant idea is that the RNA tells the assembly line how to make a protein. And of course, a protein is the antigen that the immune system can see to identify a foreign protein and develop antibodies against it. And what's not generally known, that in farmer vaccines, where they would take the entire length of a virus or a bacteria that there are multiple segments of nucleic acid in there, for example, in the virus, to allow the virus to know how to transfer across the membrane inside the cell, and then instructions of how to take over the ribosome, the endoplasmic reticulum, and the Golgi, and get the cell to start making viral proteins and viral material instead of that which originally the cell was made for. Well, these whole virus vaccines, which were much more effective and actually prevented infections, also had a low incidence of actually causing the disease. For example, smallpox, which has an attenuated virus, in a certain percent, very low percentage, actually the people get the smallpox. And there's even a lower percentage that can die from actually receiving the vaccine. So the idea was to take a RNA segment that codes for how to make the protein antigen that's in the vaccine, and never expose the host or the individual to the entire vaccine complement. And that way you would avoid anybody ever getting the disease, because you just make that one segment. Well, unfortunately, the fragment of the virus that the RNA is made from doesn't give you complete protection. And in fact, as we'll get into here in a minute, there's something else at work that caused a paper to be published that explained to me that I'm trying to get across to you on why this vaccine has been a catastrophe.
Sorry, I didn't know I was muted. Thank you for telling me, Jim. So we're gonna take a break right there, and we'll complete this next time. But I do really appreciate Dr. McDaniel sharing these facts with us. He is a scientist, a doctor. He's sharing facts and information.
Hello?
Can you hear me? Am I still muted, guys? Can you hear me, Dr. McDaniel? Uh-oh. Are we having trouble again? Trouble in paradise over here. You still can't hear me. Oh no. You can hear me, Jim. Okay. That's really weird. Maybe it is delayed. Now Dr. McDaniel is muted. Can't unmute your guest because they chose to mute themselves.
Can you hear me?
Yes, I can hear you now.
Has it been silent for about 15 minutes?
No.
You've been getting sound.
Yeah.
All I've had is silence.
Okay. Yeah, we're having a little bit of, a little bit of trouble. We're talking about things they don't want us talking about. So I'm going to be brave and leave this up and see how it goes. I did demonetize the video, so hopefully that gives us a little more edge to try to get information out there. Once again, I was just trying to let everyone know that you are a scientist and a doctor, and that you're sharing facts from studies and information. You're sharing just science that's taught in school about how a cell works, and just facts.
Yeah.
So we'll kind of go from there, and we'll hope for the best that we can continue to share these facts and information. I did have one question from a friend in the chat, and he's asking about blood pressure, and asked if magnesium was a good alternative for helping with blood pressure, or how that affects the body. Does the body respond well and help itself with if you have high blood pressure, or is New Eden a good choice for high blood pressure versus taking benzapril? I haven't heard of that.
I could respond to that by saying that I still remember in junior medicine, we were asked about the mechanisms for high blood pressure, and there were about 15 different causes of high blood pressure. I had a PhD come to me and bring me a book that he had high blood pressure, and he took magnesium, and it totally corrected his high blood pressure. He wrote a whole book on it, and of course, he just felt like everybody should know about this, but that's not. Magnesium is not the cause for everyone having high blood pressure.
Right.
It has multiple causes, and I was shocked when I traveled and did lectures into the Virgin Islands because the people, the man on the street, tend to be pure Africans. They used to work in the sugarcane fields, and they're not mixed with the Caucasians like most of our Afro Americans in the United States are. They're pure Africans, and they had responses, particularly to high blood pressure, which is very common coming into a Western society and what our food is. And they could take the ingredients in New Eden, and they got wonderful responses to lower their blood pressure. And in fact, every deviation from health that these pure Africans had responded to more comprehensive nutrition.
Wow.
And so there is not one thing that you can do that will cover all causes of high blood pressure. There's some people that the beet powder and the fresh beets seems to help their high blood pressure more than any other thing. I don't know what it is in beets that they need, but I take 2 high blood pressure medications each night to try to keep my blood pressure down. Whereas I have met people that just taking the active ingredient from the aloe, ace-mannin, will lower their blood pressure. And it's a complex issue with no one solution. I can put it that way. And this is one of the things that's interesting about nutrition is that our genes, we're told we have about 30,000 genes. And I showed the picture on there of the assembly line that is in every cell that you can see by electron microscopy. That is 3 major steps: the ribosome, the endoplasmic reticulum, and the Golgi. And the nutrient molecules are brought in by the transport RNA remarkably. I'm not sure that you followed this, but when the RNA is made from the DNA, it's made into 2 types of RNA. One type goes to the assembly line that gives the instructions of how to put together the different nutrient units, whether it be an amino acid, a fatty acid, a carbohydrate, or a mineral, or a phytochemical, how you hook them together. The transport RNA goes and is processed in the nucleolus of the cell, and it goes out to find in the fluid of the cell and in the interstitial fluid between cells the nutrient molecules that will be called for by the genes.
Okay.
And I mentioned there are 30,000 genes. It depends on which gene is activated, whether it's going to make an enzyme or
Give the instructions. Is that like I was watching Bruce Lipton, and he talks about epigenetics and how our DNA, we always thought it was our DNA had like this one instruction pattern. And then he said, but then in the lab, we were proving something different because we would take that same tissue, DNA tissue, and we would put it in different environments. And depending on the environment, it would grow different things. It might grow bone in one dish, tissue in another, and uh, let's say flesh or fatty cells in a different, because it was the message or the environment that it needed uh, to give the cells the instructions of how to what to create with those cells.
Well, you mentioned epigenetics. For
Hmm.
decades, it was believed, kind of like the tyranny of the genes, kind of like a piano, a keyboard, that
Hmm.
if you get it properly tuned, that middle C, the vibrations and the sound of it in Fort Worth, Texas, is just exactly like it is in Moscow when they have these super piano competitions.
Hmm.
Well, what they found out with epigenetics is that you look at this piano keyboard, you can play classic music on it, or you can play jazz, or you can play improvisational jazz, or you can play rock music, depending on how you hit them and how you hold them and how you combine them. Well, epigenetics is more like the fact that the genes are not just set, that even twins, when they're born, have nearly exact genes. But the longer they live, and particularly if they're not live, don't live in the same house, but get separated and have a different diet over the years and different experiences over the years, their genes change, and they call this epigenetic change. And it's like a keyboard that you think is fixed. However, depending on how your uh, molecules in your body work with them and what your experiences are, that it can modify the expression of the gene. And that's what epigenetics is about. And this is one of the reasons that if you stick around with me very long, I'll start telling you that your mind, which is the function of the brain, influences all the systems of your body. And you need to be very careful about what you believe and what you think, and particularly to think positively and in more toward the future rather than digging in the pain of the past. And as I say, that life is kind of like you bring in fresh food, but over a period of time, it becomes garbage, and you got this garbage can on your back. And garbage doesn't smell good, it doesn't taste good, it doesn't look good. But if you move forward, there's always fresh new food, and you can throw it over your shoulder in the garbage can and move forward, thinking of the future and positively, and ignore the things of the past and enjoy the possibilities and the potential of the future. So, what you're thinking about and what you believe can be very powerful. And I have people come to me, oh, I've been going to some doctor, so and so. I think he's a numbskull. He doesn't help me, and done any good. And I look at him and I say, do you have confidence in your doctor? And they say, no. I said, well, you need to find you a doctor that you do have confidence in and you believe in, and you will do a whole lot better. And this is the truth. It's the mind influences all the systems, and that's part of the epigenetics of how the genes can be played differently, just like a keyboard on the piano. I hope that's an explanation of that. But
That is, it's great, and that's so helpful because life really is what you make it. Where we put our energy and our focus is what grows, and that's such a strong statement, and so much a part of what we believe here and what we focus on, and so that's where your thoughts and values are so in line with what our community believes as well. So, and I love the product New Eden. Our whole family takes it. My husband puts his in his workout shake every morning. I take mine at night. And my little boy takes his gummies that we make for him. And And we always take it on a full spirit, with a full spirit. So I advise everybody to check it out. We'll be back with Dr. McDaniel again to finish this discussion, this brave discussion on facts and not politicizing science. There is no room for politics in science and people's health and wellness. We're just here to state the facts. So thank you, Dr. McDaniel. Do you have any closing statements before we go?
Well, I wanted to say that one of the questions I saw that was asked is how long is it in the blood, and
Oh,
how about taking it? Well, it's good to take it two or morning and night. And we have some people that take only in the morning and some at night, and they do well. And we have people that take it morning, noon, and night.
yeah.
But in tissue culture, we found out that the most important nutrient, which is the ace mannan that's in the aloe leaf gel, where so much money was spent by Carrington Laboratories trying to make a drug of what we eventually found out was a nutrient, this ace mannan, that in tissue culture, the tissue culture cells would take it up, and inside of them, and you could stain it with special stains to see that it's inside the cells for 5 to 7 days as enzymes begin to uh, click off or clip off the different sugars in the long chains. So there is an inherent time release system in the ace mannan that probably if you missed a dose today that you didn't take yesterday, you still got it on board because there's a time release mechanism in nature that is in ace mannan. And I can't tell about all the other different molecules that are made in plants, but
Mhm.
this is, as I started out in this, trying to tell you as we went through all this detail, that there's incredible information known about how detailed and engineered the activity of the cell is, and that for people to start saying that it operates by just how you feel is very superficial. Like I started out with, that the XX chromosome is that of a female and
Yeah,
an XY is that of a male. It has been and will continue to be.
just
And
has.
I heard an expert on Sunday on Meet the Nation pointing out that these people that are having gender dysphoria type things, that it's less than 1% that all this hullabaloo is going on about people feeling like they will. Well, I'm a boy that's in a girl's body, or I'm a male that actually internally I'm a female. And probably a lot of these people have emotional problems. We have very poor parenting and home life now in our country. It's the truth that virtually a new couple, it takes both of them going out to work in our economy to be able to provide a home and everything that you require to have a household. And so, mothering and taking care of children that used to be probably 90% of the families is now a small percent. And so, we're not having generations that identify with their mother or their father that matches to their genetic makeup. And we have a lot of dysfunction in our society because of different forces. And some people don't know it, but in the 1980s, I was a elected member to the Texas State Board of Education. And I found out that our public schools were being used to present a curriculum that attacks all traditional values, everything that made the United States a wonderful country, everything that made our homes solid and strong, with parenting. All of this is being attacked by some of these political agendaed people and forces and groups. And we need to get back to traditional values and to the truth and facts, which I'm trying to show how incredible the engineering and design of cellular life is, and that for somebody just all of a sudden come up with a hot idea totally out of sync with nature is fallacious.
Absolutely. And I just want to mention this really quick before we go. I know I've got to run as well, but you mentioned the tie, and I want to get into this more next time you're on, talking about how they're tying this with the green energy deal. This was a tweet that was from. They're talking about this is a congressional record transcript from 2015, and they are saying that the green energy mandates have required the Army and Navy to install solar arrays at various facilities. At Naval Station Norfolk, for example, the Navy spent over 21 million to install a 10-acre solar array, which will supply a grand total of 2% of the base's electricity. According to the Inspector General's office, this project will save enough money to pay for itself in just 447 years. Too bad solar panels only last 25 years. So this is and there's a whole report on this that I can bring up next time that we meet, once we get into that part of your video, or after the video, once you have address it. But they've known this has all been known, that this is a false narrative that they're shoving down our throat. They want to. I tell my little boy when he tries to pull one over on me, you can act like you're stupid, but don't try to act. Don't ask me to act like I'm stupid. And that's what they want us to do with all of this. So I'm so proud to be standing with you in truth in this. And we'll take it on the chin and see what happens. So thank you again for joining us, Dr. McDaniel. You guys, please give us a like, share the video. Let's see what happens. Let's get some truth out there. Take care.
Thank you.